Have you ever stopped to think about the silent, really intricate biochemical pathways that are constantly working inside your cells?
Hmm.
I mean, they're not just about churning out energy, right?
At all.
They're involved in building, protecting, even communicating, basically sustaining life itself.
Yeah.
It's easy to just think of our bodies as, muscles and bones.
Right. The big picture stuff. Yeah.
Exactly. But underneath all that, there's this astonishing hidden universe of molecular machinery.
It really is incredible.
And today, we're not just scratching the surface. We are going to embark on a true deep dive into one of the most, I think, fascinating and foundational of these pathways.
That's right. This deep dive is all about the pentose phosphate pathway. You might also know it as the hexose monophosphate shunt.
Right, H&P shunt.
Yeah, exactly. And it's absolutely vital product.
Yep.
Nicotinamide adenine dinucleotide phosphate, which thankfully we can just call NADPH.
Much easier.
Definitely.
Yeah.
Think of them as kind of the unsung heroes of cellular resilience and construction.
And when we say vital, we really, truly mean it. The significance of this pathway and this one molecule, NADPH, it really can't be overstated.
No, it's fundamental.
We're talking about the core ways your body defends itself against constant damage, how it synthesizes these essential compounds that are literally the fabric of life, and maintains critical functions that are, well, life-sustaining. No exaggeration.
Absolutely.
So our mission for you today, as we meticulously unpack our source material, isn't just to, understand this complex information. It's really to grasp it with the depth and insight of an elite professional in this field. You'll truly see the inner workings of cellular mastery.
Exactly. We're going to guide you through each concept, each connection, making sure it's not just crystal clear, but that you get a holistic understanding. Not just what these processes are,
Right.
but why they are absolutely critical for life, how they interconnect with everything else metabolically, and maybe most powerfully, what happens when things go wrong. We want to bring these microscopic processes to life, connect them directly to your health, your well-being. So let's begin this journey into this really remarkable metabolic diversion.
All right, let's unearth this biochemical marvel.
Yeah.
We're starting with a kind of foundational overview of the pentose phosphate pathway itself. So first things first. Where does this all happen inside our cells? And what makes it so unique compared to the big players everyone talks about, like, say, glycolysis or the PCA cycle?
Excellent questions to kick us off. So the pentose phosphate pathway, or PPP as we'll probably call it,
Yeah, PPP.
it takes place entirely in the cytosol of cells.
Okay, so the main fluid part of the cell.
Exactly, right there in the cellular soup, outside the organelles. Now, what really sets it apart, and this is a key difference, is that it does not directly consume or produce any ATP.
No ATP at all. That's unusual.
It is. Think about it. Most major metabolic pathways are either burning ATP or making it. The PPT is entirely ATP neutral. And that immediately tells you its main job isn't generating the cell's energy currency, it's doing something else. Something equally, maybe even more fundamental.
Okay, that's fascinating. So if it's not making ATP, what is its primary purpose? And you mentioned earlier it has two distinct phases. How do those work?
Precisely. The pathway is elegantly split into two very distinct phases, each with its own job. First up is the irreversible oxidative phase.
Irreversible. Okay.
Yeah, think of this as the initial kind of one-way street part of the pathway. Once a molecule enters here, there's no going back.
Right.
This is where the real magic happens for NADPH production. During this phase, something quite specific occurs. Carbon number 1 of a glucose 6-phosphate molecule is actually released as carbon dioxide.
CO2, really, like in the TCA cycle.
Exactly, yes, CO2 is released. And at the same time, it produces a 5-carbon sugar phosphate, specifically, ribulose 5-phosphate. And critically, it generates 2 molecules of NADPH.
2 NADPH, okay.
This is the powerhouse section for creating those vital reducing equivalents we'll be talking so much about.
2 molecules of NADPH per glucose 6-phosphate. That's quite a yield from a pathway that doesn't even touch ATP directly.
It is.
And then what about the second phase? You described it as reversible. What does that mean for the cell? More flexibility?
Yes, exactly. The reversible non-oxidative phase. This is the adaptable two-way street of the pathway. Unlike the oxidative phase, its rate and direction are really flexible.
Okay.
They dynamically adjust based on what the cell needs right then and there. It's metabolic supply and demand for the pathway's intermediates. It's basically a sophisticated metabolic interchange.
Like a railway junction.
Yeah, that's a great analogy. A complex railway junction where different sugar molecules with anywhere from 3 to 7 carbons can be interconverted, shuffled around, redirected depending on the cell's immediate needs. It's all about metabolic agility.
So we've got these two phases working together. What are the core functions that come out of this intricate molecular choreography? You mentioned NADPH and those pentose sugars.
That's right. At its heart, the pentose phosphate pathway serves 3 major, absolutely indispensable functions. First, and maybe foremost, it's the major source of NADPH in the body.
The main source.
The main source. Remember, NADPH is this essential biochemical reductant. It's crucial for electron-donating reactions that drive synthesis building things and cellular protection, like a molecular antioxidant defense team.
Okay, and the second function.
Second, it's responsible for ribose 5-phosphate production. And this isn't just any sugar. It's a foundational molecule, absolutely required for making nucleotides.
The building blocks of DNA and RNA?
Exactly. DNA, RNA, and even ATP itself. You literally cannot replicate cells, repair DNA, or make basic energy currency without it, without ribose 5-phosphate. Well, life as we know it just stops.
Wow. Okay, so NADPH for protection and building, ribose 5-phosphate for genetic material and energy. What's the third major function? Is that where the shunt part of its name comes in?
Indeed, that's precisely where the shunt idea comes in. It really highlights how interconnected this pathway is. The third function is glycolytic intermediary conversion.
Okay.
Meaning the PPP provides this brilliant mechanism to convert those pentose sugars it produces, or maybe other sugars, with 3 to 7 carbons that might be in excess,
right,
back into intermediates like triose and hexose phosphates. Things like fructose 6-phosphate and glyceraldehyde 3-phosphate.
Which can feed right back into glycolysis.
Exactly. They can feed directly into glycolysis, that central pathway for energy generation. This shows its role as a shunt, seamlessly integrated with other major metabolic routes. It gives the cell incredible flexibility in using its sugar resources. It's truly a central hub on the metabolic map, a master coordinator of carbon flow.
Let's really dive deeper into those irreversible oxidative reactions then, because that's where the critical NADPH gets generated. What's the starting point, and can you just reiterate the specific outputs and why they matter so much?
Absolutely. So each molecule of glucose 6-phosphate that enters this oxidative phase is the key starting point.
Okay.
Through a series of steps, it gets oxidized. This leads to the formation of ribulose 5-phosphate, that vital pentose sugar we talked about, along with a molecule of CO2, specifically from carbon 1 of the original glucose.
Right.
And most importantly, we get those 2 crucial molecules of NADPH.
2 NADPH. Got it.
This part of the pathway isn't just busy, it's particularly vital in tissues that have an extraordinarily high demand for NADPH. It acts like their dedicated power station for all these reductive processes.
And where exactly is that demand highest? Can you give us some concrete examples? It helps to picture its importance in the body.
Sure. Think about processes that involve a lot of building, a lot of molecular construction. For instance, in metabolically active tissues like the liver or lactating mammary glands, and adipose tissue are fat cells.
Okay.
This phase is absolutely critical for fatty acid biosynthesis. Building huge amounts of lipids for energy storage or milk production relies heavily on NADPH.
Makes sense.
Then in steroid-producing tissues, think testes, ovaries, placenta, the adrenal cortex, it's essential for steroid hormone biosynthesis, another complex building process needing NADPH's reducing power.
Hormones too, okay.
And here's a really critical one for defense. In red blood cells, RBCs, it's vital for the NADPH-dependent reduction of glutathione.
Ah, the antioxidant connection.
Exactly. Glutathione is the frontline defense against oxidative damage in red cells. This foreshadows its absolutely critical role in maintaining red cell integrity, protecting them from constant threats. So it's a pathway for both sophisticated building and robust protection.
So whether it's building vital molecules or standing guard against damage, the PPP is central. Okay, let's get into the nitty-gritty of that first pivotal step. Glucose 6-phosphate dehydrogenation. I understand this isn't just any step, it's the rate-limiting one. Why is that so significant?
It is indeed the critical step, and it's catalyzed by a key enzyme called glucose 6-phosphate dehydrogenase, or G6PD.
G6PD.
This enzyme performs the initial pivotal reaction. It oxidizes glucose 6-phosphate to 6-phosphoglucosilactone. And in doing so, it reduces NADP plus to NADPH.
Okay.
This is hugely significant because G6PD isn't just a catalyst. It's the committed, rate-limiting, and highly regulated step. Think of it as the ultimate gatekeeper for the entire pentose phosphate pathway.
Committed, rate-limiting, and highly regulated. What does that really mean for how the cell controls this pathway? Why have such a strict gatekeeper?
It means it's the bottleneck. It's the primary control point. If you want to control the flow of all the traffic down this pathway, you control G6PD.
Makes sense.
If this enzyme is slow or inhibited, the whole pathway slows down dramatically. Conversely, boost its activity, and the pathway speeds up. This control is crucial because the cell needs to precisely balance its production of NADPH and ribose 5-phosphate with its immediate needs.
So how is it regulated? How does the cell exert that control?
Well, one of the most important and, frankly, elegant mechanisms is NADPH feedback inhibition.
Ah, the product inhibits the enzyme.
Exactly. NADPH itself acts as a potent competitive inhibitor of G6PD. Imagine NADPH essentially saying, hey, we've got enough reducing power here, slow down.
Right.
If the cell has plenty of NADPH, meaning the NADPH-NADP plus ratio is high, this high ratio actually binds to G6PD and tells it to slow down. The cell doesn't need more reducing equivalents right now.
Like a thermostat.
Precisely.
Yeah.
It's like a thermostat for cellular reductive power. Conversely, if that ratio decreases, meaning NADPH is being used up quickly, indicating high demand, this inhibition is relieved.
Okay.
And the flux through the pathway significantly increases. It's a sophisticated, self-adjusting feedback loop, ensures the cell makes exactly what it needs, no more, no less.
That's a very clever internal system. Are there any external signals, like hormones, that also influence G6PD?
Yes, absolutely. Another important regulator linking this pathway to broader metabolic states is insulin.
Insulin, okay, how?
Insulin actually upregulates the expression of the gene for G6PD.
Ah, so it tells the cell to make more of the enzyme itself.
Exactly. So in the absorptive or fed state, basically,
Yeah.
after you've eaten and your body's in building mode, anabolic mode,
Right.
insulin signals to increase the amount of G6PD enzyme. This leads to increased pathway activity, making sure the cell has plenty of NADPH for all those synthetic processes like building fatty acids or cholesterol. It's a perfect example of the body anticipating metabolic needs.
Okay, so that's the first highly controlled gatekeeper step. What happens to the 6-phosphoglucanolactone that's formed next in this oxidative journey?
Good question. Following that initial step in the second reaction, the 6-phosphoglucanolactone is rapidly hydrolyzed, basically, split by adding water by an enzyme called 6-phosphoglucanolactone hydrolase. A fairly straightforward step.
Yeah.
Then we move to the third step, which is another crucial one, an oxidative decarboxylation of the product, 6-phosphoglucanate.
Oxidative decarboxylation, meaning it loses the CO2 and gets oxidized.
Precisely. This reaction is catalyzed by 6-phosphoglucanate dehydrogenase. And this is where the second molecule of NADPH is generated,
Ah, the second one.
doubling our reductive power from this phase. Plus, we get ribose 5-phosphate, that vital pentose sugar phosphate, and the final molecule of CO2, specifically from carbon 1 of the original glucose.
So, by the end of this irreversible phase, 2 NADPH, 1 ribose 5-phosphate, and 1 CO2.
You got it. All ready for the next steps or other cellular demands?
Yeah.
A highly efficient little factory.
Now that we've powered up the cell with NADPH, let's shift focus to the reversible non-oxidative reactions. You said this phase is like a metabolic interchange, super flexible. Where do these reactions happen, and what's their overall importance?
Right. These reactions are pretty much ubiquitous. They occur in virtually all cell types.
Everywhere.
Pretty much everywhere, yeah. But they're particularly crucial in cells that are actively synthesizing nucleotides and nucleic acids. Think about any cell undergoing rapid growth or division. They absolutely depend on these processes. They are fundamental. And the interchange analogy is spot on. These reactions shuffle around sugar molecules containing anywhere from 3 to 7 carbons. It gives the cell immense metabolic adaptability. Like having a versatile set of Lego bricks, you can reconfigure into whatever shape is needed right now.
So the cell really decides where the ribose 5-phosphate goes based on its needs. What are those different fates? How does demand dictate direction here?
Exactly. The fate of ribose 5-phosphate isn't fixed. It depends entirely on the cell's immediate needs. Let's look at 2 main scenarios because it highlights the pathway's intelligence.
Okay, scenario 1.
Scenario 1. Imagine a cell that's rapidly growing, dividing, or maybe repairing its DNA extensively, like an immune cell fighting infection, or a cell in an embryo.
High demand for building blocks.
Exactly. This cell has a very high demand for nucleotides, meaning it needs more ribose 5-phosphate than it might need NADPH at that moment.
Right.
In this case, The non-oxidative reactions can directly and efficiently convert ribulose 5-phosphate into ribose 5-phosphate, providing those essential building blocks for DNA, RNA, ATP, right when they're needed most.
Okay, makes sense. And what if the cell's needs are flipped? What if it needs tons of NADPH for, say, synthesis, but isn't so hungry for ribose 5-phosphate?
That's scenario 2, and it really shows the PPP's metabolic economy. When a cell needs a lot more NADPH for reductive biosynthesis, maybe it's a liver cell making loads of fatty acids.
Right.
But it doesn't need an excess of ribose 5-phosphate. The pathway cleverly shunts the ribulose 5-phosphate into intermediates of glycolysis.
Back into glycolysis, how?
It converts them into fructose 6-phosphate and glyceraldehyde 3-phosphate. This means the PPP can provide that crucial reducing power, the NADPH, without overproducing nucleotide precursors it doesn't need right now.
That avoids waste.
Precisely. And the clever part is, those glycolytic intermediates can then be either burned for energy or used for other things. It's a beautiful example of adapting metabolic flow to exact demands.
That flexibility is truly remarkable. What are the key enzymes doing all this sophisticated shuffling? And do they need any special cofactors we should know about?
Yeah, there are 2 particularly important enzymes here, kind of like master carbon skeleton rearrangers. The first is transcatalyase.
Transcatalysis, okay.
It functions as a 2-carbon unit transfer enzyme. It basically picks up 2 carbons from one sugar and sticks them onto another. And crucially, this enzyme absolutely requires thiamine pyrophosphate, TPP, as a coenzyme.
TPP, that comes from vitamin B1, right?
Thiamine, that's the one, vitamin B1 derivative.
Mm-hmm.
The second key enzyme is transoldolase.
Transoldolase.
And this one transfers 3 carbon units between sugars. Again, just precisely rearranging those carbon backbones.
Thiamine pyrophosphate, TPP, sounds incredibly important if transcatalyase needs it. What's its broader significance? Does a thiamine deficiency mess up other things besides the PPP? What would that mean?
Oh, it's incredibly important. And yes, a thiamine deficiency, meaning low TPP, would cause widespread and severe metabolic problems.
Why is that?
Because TPP isn't just for transcatalyase. It's required by several other absolutely vital multi-enzyme complexes across different central metabolic pathways.
Yeah.
Think of TPP as a keystone cofactor for critical metabolic junctions.
Like what else?
Like pyruvate dehydrogenase, the crucial gateway linking glycolysis to the TCA cycle, basically turning sugar breakdown products into fuel for the main energy factory.
Okay, huge.
Huge. It's also needed by ketoglutarate dehydrogenase, a key enzyme within the TCA cycle itself, central for energy generation and making building blocks. And also by branched-chain ketoacid dehydrogenase, vital for breaking down certain amino acids.
Wow. So if you're low on thiamine.
If you're low on thiamine, not only does your PPP suffer impairing antioxidant defense and nucleotide synthesis, but these other central pathways get severely hit too.
Right.
This can lead to significant systemic health issues like beriberi or Wernicke-Korsakoff syndrome. It really underscores how interconnected everything is. One missing piece ripples through the whole system.
Okay, so that covers the pathway itself pretty thoroughly. Now, let's shift our focus entirely to its most famous product, NADPH, the body's master reductant. What makes NADPH so unique? I mean, it sounds an awful lot like NADH, which we hear about more often with energy production. What's a subtle difference that matters so much?
That's a sharp observation.
Yeah.
And it gets right to a subtle but profound structural difference that dictates their totally different metabolic roles. NADPH differs from NADH by just a single phosphate group attached to one of its ribose units.
Just one phosphate group, that's it.
That's it. A tiny molecular tag, but it changes everything.
How does that tiny change work functionally in the cell?
It allows for exquisite enzyme specificity and distinct roles. That little structural tweak means NADPH interacts almost exclusively with NADPH-specific enzymes.
Ah, so different enzymes recognize it.
Exactly. These enzymes perform unique functions, different from those using NADH. Think of it like a key and lock. The phosphate group on NADPH is a special groove that only fits certain locks, the NADPH-dependent enzymes. NADH, without that groove, fits entirely different locks, different enzymes.
So NADH is more for breakdown, energy harvesting.
Primarily, yes.
Yeah.
NADH is mainly involved in catabolic reactions, collecting electrons often to make ATP via the electron transport chain, like collecting energy from fuel.
And NADPH.
And NADPH is built for anabolic or synthesis reactions. It donates electrons to build complex molecules. It's like supplying the power tools and materials for construction.
And what about their levels in the cell? Do the ratios reflect these different jobs? That would be pretty neat.
They absolutely do. It's a beautiful illustration of cellular strategy. If you look at their redox ratios in the cytosol of, say, a busy liver cell,
Okay.
the NADP plus NADPH ratio is remarkably low, maybe around 0.1.
Low, meaning lots of NADPH.
Exactly. It strongly favors the reduced form, NADPH. There's always a high concentration of NADPH ready to donate electrons for building things, like a continuously loaded gun of electrons for synthesis.
Okay. And NADH.
In stark contrast, the NAD plus NADH ratio is incredibly high, maybe around 1,000.
1,000. Wow. So mostly NEG plus dirts.
Right. Strongly favors the oxidized form, NAD plus TERS. This makes it ideal for oxidative catabolic reactions, where it can readily accept electrons from fuel breakdown, like a perpetually empty bucket ready to collect electrons.
So the cell keeps them separate, strategically. One pool for breaking down, one for building up.
Precisely. Strategic separation of electron carriers for distinct metabolic purposes. It's really quite elegant.
So to boil it down, the core functions of NADPH fall into what two main categories?
Primarily, they are reductive biosynthesis, literally building complex molecules and detoxification reactions, protecting the cell from harmful stuff. Those are the two big buckets where NADPH shines.
Let's unpack those diverse applications then, starting with reductive biosynthesis. How is NADPH a high-energy electron donor? And what kinds of critical molecules does it help build?
Think of NADPH as a powerful battery, specifically designed for construction projects. It's high energy because it holds on to some of the energy from that initial glucose-6-phosphate oxidation in its reduced state.
Okay.
But the critical difference from NADH is how its electrons are used. NADPH's electrons are channeled into biosynthetic pathways, building complex molecules from simpler ones. They aren't usually sent to the electron transport chain for ATP like NADHs are.
Right.
Its highly negative reduction potential makes it an excellent electron donor, readily giving up electrons to drive those energetically tough synthesis reactions forward. It's the go-to power source for building.
So it's the synthesis powerhouse. Give us some concrete examples of what it helps create, just to illustrate its range.
Absolutely. NADPH is indispensably essential for the synthesis of fatty acids. Think liver, adipose tissue,
Right.
lactating mammary glands, anywhere you need to make lots of fat for storage or milk. That all depends on NADPH.
Huge amounts of fat. Yeah.
It's also crucial for cholesterol synthesis in the liver, which is the precursor for so many other vital molecules.
Like steroid hormones.
Exactly.
Okay.
And that's another big one. It powers steroid hormone synthesis in organs like the placenta, ovaries, testes, and adrenal cortex. All these complex, vital biomolecules, from energy storage fats to regulatory hormones, rely heavily on NADPH's reducing power. Without it, you couldn't assemble these building blocks of life.
Beyond building, you mentioned detoxification as the other major role. Let's talk about hydrogen peroxide reduction. How does NADPH fit into the body's antioxidant defense system? First, what are reactive oxygen species, ROS, and why are they such a threat?
Right. Reactive oxygen species, or ROS, things like hydrogen peroxide, H2O2, and superoxide radicals, O2S, are incredibly reactive compounds. They're formed continuously inside us.
Just from normal metabolism.
Yeah, partly as byproducts of normal aerobic metabolism, kind of like the exhaust fumes from our cellular engines. But their levels can also shoot up due to exposure to certain drugs, environmental toxins, or even if our own antioxidant defenses are low.
And when they build up,
When these reactive species accumulate, they create a dangerous state called oxidative stress. These things are highly reactive and can cause severe, indiscriminate chemical damage. They damage DNA, messing with genetic info. They damage proteins, impairing function. They damage unsaturated lipids, destabilizing cell membranes.
that's bad.
It is. If it's unchecked, it can lead to cell dysfunction or even cell death.
And that kind of damage sounds like it would have broader health implications, right? Disease connections.
Profoundly serious, yes.
Ah,
The implications are far-reaching. ROS are implicated in many severe pathological processes. Things like the damage and reperfusion injury after a heart attack or stroke,
okay.
the development and progression of many cancers, various inflammatory diseases, and even the fundamental processes of aging itself. It's a constant silent battle inside us. And it's worth noting, sometimes our own white blood cells deliberately generate ROS to kill microbes, kind of like controlled friendly fire. But in most contexts, ROS are a major threat the body needs to manage very carefully.
So, given this constant threat, how does the body handle it? What are the key enzymatic defenses? And crucially, where does NADPH come in?
The cell has evolved several clever protective mechanisms. One of the most critical involves an enzymatic defense team. And a key player here is reduced glutathione, or GSH.
Glutathione.
GSH is a small tripeptide thiol found in almost all cells. It acts as a critical intercellular chemical detoxifier, especially for hydrogen peroxide.
How does it work?
An enzyme called glutathione peroxidase, which needs selenium to work, it's a selenoprotein, catalyzes the reaction. It uses GSH to neutralize H2O2, converting that harmful peroxide into harmless water.
Okay.
But in doing so, the GSH itself gets oxidized, forming oxidized glutathione, or GSSG. And in this oxidized form, it can't protect anymore. It's lost its electron donating power.
So it needs to be regenerated. And this is where NADPH steps in, right? To recharge the glutathione.
Precisely. This is where NADPH's role becomes absolutely indispensable. The cell can't afford to run out of active GSH, so it needs to constantly regenerate it from the oxidized GSG.
Oh.
This vital regeneration is catalyzed by the enzyme glutathione reductase, and it uses NADPH as the crucial source of reducing equivalents, those high-energy electrons, to convert GSSG back to two molecules of active protective GSH.
So NADPH doesn't touch the peroxide directly.
No.
But it powers the regeneration of the molecule that does.
Exactly. It indirectly provides the essential electrons for H2O2 reduction, making it absolutely indispensable for antioxidant defense. Without a continuous supply of NADPH, the cell's ability to detoxify hydrogen peroxide would just collapse.
Like a relay race.
Yeah, a perfect analogy. NADPH passes a baton to glutathione reductase, which recharges glutathione, allowing it to quench the oxidative threat. And it's not alone. Other enzymes like superoxide dismutase and catalase also help neutralize ROS, forming a comprehensive, layered defense system.
Beyond these enzymes, what about antioxidant chemicals? You know, the ones we hear about in food and supplements, vitamin C, vitamin E, beta-carotene. How do they fit in?
That's a really interesting and often misunderstood area. There are indeed several common intracellular reducing agents, ascorbate, vitamin C, vitamin E, carotene, that are known to detoxify ROS in lab settings in test tubes.
Right.
And it's true that eating foods naturally rich in these compounds consistently correlates with a lower risk for certain cancers and other chronic health problems. So it's very tempting to assume it's directly because of their antioxidant effect in the body.
Ooh, there's a catch, isn't there? Something about clinical trials using isolated supplements giving surprising results.
This is where it gets really interesting, and it's a crucial aha moment. Despite that strong correlation with dietary intake, clinical trials using isolated antioxidant compounds as dietary supplements have largely failed to show clear beneficial effects in preventing disease.
Really? They don't work as expected?
Largely, no. And in some cautionary cases, there have been unexpected, even negative outcomes. A classic striking example is carotene supplementation. In large trials, high doses actually increase the rate of lung cancer in smokers rather than decreasing it.
Wow, the opposite effect. That's genuinely counterintuitive.
It was a profound and unexpected finding.
So what's the big takeaway then? Are supplements useless, or is it more complicated?
The profound takeaway here seems to be that the health benefits of fruits and vegetables are likely due to a highly complex interplay between many naturally occurring compounds, not just one or two specific antioxidants, acting synergistically within the intricate environment of the whole food in the body.
So it's the combination, the whole package.
It seems so. Our understanding is still evolving, but it's rarely as simple as take this one isolated compound to fix a complex biological issue. Nature's solutions are often far more integrated than our isolated supplements can replicate.
That's a powerful insight. Okay, let's move to another critical role for NADPH. The cytochrome P450 monooxygenase system, or CYP system. What are monooxygenases, and how is NADPH involved in this incredibly versatile system?
Right, the CYP system. Monooxygenases, sometimes called mixed function oxidases, are enzymes that do a very specific vital reaction. They take one atom from molecular oxygen, O2, and incorporate it into a substrate, usually forming a hydroxyl group adding an O handle.
Okay.
And simultaneously, they reduce the other oxygen atom to water. And here's the key part for us. In the cytochrome P450 system, NADPH provides all the necessary reducing equivalents, all the electrons, for these crucial reactions. Its role as the sole electron donor here is absolute.
Got it. NADPH fuels the reaction, and the P450 name.
Oh, that just refers to their characteristic light absorption peak
Yep.
for 450 nanometers when they're bound to carbon monoxide. It's a technical detail, a spectral signature. They're actually a huge superfamily of related heme-containing enzymes with incredibly diverse roles.
So where do we find these versatile CYP enzymes, and what are their distinct jobs? I think you mentioned two major systems.
That's right. Two distinct systems, different locations, different primary functions. First, there's the mitochondrial system.
In the mitochondria, the powerhouses.
Exactly. Associated with the inner mitochondrial membrane. Its main job is absolutely crucial for biosynthesis of steroid hormones. In those steroidogenic tissues you mentioned, placenta, ovaries, testes, adrenal cortex, it precisely hydroxylates intermediates in the complex pathway, turning cholesterol into steroid hormones like cortisol or testosterone.
And that hydroxylation, adding the OH group, makes them more water-soluble.
Precisely. It's vital for making these normally hydrophobic, fat-soluble molecules more water-soluble so they can be transported and eventually excreted more easily.
Does this mitochondrial system do other things too?
Yes, it's quite versatile. The liver uses the same system in the early steps of bile acid synthesis, which are essential for digesting fats. It's also critical in activating vitamin D. It hydroxylates cholecalciferol, vitamin D3, to 25-hydroxycholecalciferol in the liver. And then the kidney uses a similar mitochondrial system for the final activation step to the biologically active 1025-dihydroxylated form.
So hormones, digestion, bone health. Quite a range.
Definitely. And in this mitochondrial system, the electrons flow in a specific path from NADPH to FAD, then through an iron-sulfur protein, and finally to the CYP enzyme itself to complete the reaction.
Okay, what about the second major CYP system? I hear it's really important for dealing with foreign stuff.
That's the microsomal system. This one is largely associated with the membrane of the smooth endoplasmic reticulum, and it's especially abundant in the liver, our body's main detoxification center.
Makes sense.
Its primary, incredibly vital function is the detoxification of xenobiotics.
Xenobiotics, foreign compounds, right? Things not normally found in the body.
Exactly. Any chemical found in an organism that isn't normally produced or expected to be there. This includes a vast array of drugs we take, petroleum products we might be exposed to, pesticides, all sorts of foreign compounds we encounter.
So it's our body's chemical processing plant for external substances. How does it actually detoxify them? I've heard of a two-phase process.
That's generally how it works, yes.
Right.
A brilliantly coordinated two-phase approach to neutralize and eliminate these potential toxins. In phase I, CYP enzymes, like the very famous CYP3A4, perform that initial hydroxylation on the xenobiotic.
Adding the OH handle again.
Right. And this serves two crucial purposes. It can either activate or inactivate a drug, which is huge for pharmacology.
Right.
And crucially, it makes the potentially toxic compound more water-soluble, making it easier to get rid of later. It's like adding a handle or a tag to the molecule.
And phase II.
Then, frequently in phase II, that newly added hydroxyl group acts as a perfect attachment site for conjugation with a highly polar molecule, like glucuronic acid, for example.
Conjugation, meaning sticking something big and water-soluble onto it.
Exactly. This conjugation step significantly increases the compound's water solubility even further, making it much easier for the body to excrete it efficiently, usually in the urine or bile. It's like packaging the processed toxin for quick and complete removal.
That's a highly sophisticated and absolutely essential system.
Yeah.
Does it vary between people?
Yeah.
And what are the clinical implications of that? That sounds really important for medicine.
This is a critical point with huge clinical relevance.
Mm.
It really underscores the whole field of personalized medicine. We have to consider polymorphisms in CYP enzyme genes.
Polymorphisms, common genetic variations.
Yes, slight differences in the DNA code from person to person. These can lead to significant differences in how individuals metabolize drugs.
So some people break down drugs faster or slower.
Exactly. And this directly affects both drug efficacy, how well a drug works for someone, and the potential for side effects, sometimes severe ones.
Can you have an example?
Sure. Our source material mentions atorvastatin, a common cholesterol drug. Grapefruit juice contains compounds that strongly inhibit the CYP3A4 enzyme, which is the main enzyme that breaks down atorvastatin.
Right.
So if someone drinks grapefruit juice while taking atorvastatin, especially someone whose CYP3A4 might already be a bit slow due to their genetics, the inhibition can lead to dangerously high levels of the drug building up in their blood. This significantly increases the risk of side effects like muscle pain or liver damage. Just because the drug isn't being cleared properly in that individual, influenced by both genetics and diet.
Wow. That really highlights why understanding individual metabolism is so important for safe medication use.
Absolutely. It's central to pharmacogenetics and personalized medicine.
Okay, let's pivot to another fascinating and frankly dramatic role for NADPH in our immune system. How white blood cells use it for phagocytosis and literally killing microbes. How do our white blood cells engulf and destroy invaders, like tiny biological weapons?
It is pretty dramatic. Phagocytosis is the remarkable process where certain white blood cells, phagocytes like neutrophils and macrophages, actively ingest microorganisms, foreign particles, even cellular debris. It's a vital frontline defense, especially against bacteria.
Okay.
And these cells have different ways to kill what they engulf. Some are oxygen independent, but the most potent are the oxygen dependent mechanisms.
And that's where NADPH comes in.
That's where NADPH plays a crucial and starring role. Yes.
Yeah.
The oxygen dependent mechanisms are generally the most powerful bactericidal tools the body has. They involve the coordinated action of two key enzymes, NADPH oxidase and myeloperoxidase, MPO.
Okay, NADPH, oxidase, and MPO. How does it start?
It starts when an invading bacterium is recognized and internalized by the phagocytic cell engulfed into a little vesicle inside the cell called a phagolysosome.
Right.
Then NADPH oxidase, which sits in the leukocyte cell membrane, or the phagolysosome membrane after engulfment, gets rapidly activated.
And when NADPH oxidase switches on, what happens next? I've heard the term respiratory burst. Sounds intense.
It is intense, and that's exactly what happens. Upon activation, NADPH oxidase rapidly takes molecular oxygen, O2, from the surrounding tissue and reduces it to superoxide, O2.
Superoxide, a reactive oxygen species.
A highly potent ROS, yes. And while doing this, it oxidizes NADPH to NADP plus DAR. This rapid, massive consumption of oxygen that accompanies the superoxide production is precisely what's called the respiratory burst.
Like the cell is hyperventilating.
Kind of, yeah. But for a specific purpose, generating a storm of this reactive oxygen. This NADPH oxidase is actually a complex multi-protein machine on the membrane, involving a flavocytochrome and other bits. Electrons flow in a controlled way, from NADPH through FAD and heme components, ultimately to oxygen to make superoxide.
What happens if there's a problem with this vital NADPH oxidase? Are there diseases associated with that?
Yes, a very serious and debilitating condition called chronic granulomatous disease, CGD, results from rare but severe genetic defects in NADPH oxidase.
Okay.
Individuals with CGD simply can't produce superoxide effectively, or sometimes at all. This leaves them highly susceptible to severe recurrent bacterial and fungal infections. They also tend to form granulomas, these nodules of inflammation where the body tries to wall off bacteria it can't kill. It's a stark illustration of how vital this single enzyme complex is for innate immunity.
So assuming NADPH oxidase works and makes superoxide, what's the next step in killing the microbe? How does the cell escalate the attack?
Right. Superoxide itself is damaging, but the cell often takes it further. Superoxide is quickly converted to hydrogen peroxide, H2O2, another potent ROS. This can happen spontaneously or faster with an enzyme called superoxide dismutase.
Okay, so now we have H2O2 inside the phagolysosome with the microbe.
Exactly. And here's where the second key enzyme, myeloperoxidase, MPO, comes in. MPO is a heme-containing enzyme found inside the phagolysosome. In the presence of MPO, that hydrogen peroxide combines with chloride ions, Cl, which are also present, and gets converted into hypochlorous acid.
Hypochlorous acid.
Hypochlorous acid.
Okay.
Wait, isn't that? Yes. It's the active ingredient in household bleach.
Our white blood cells make bleach.
Essentially, yes. This incredibly potent substance is highly toxic to bacteria and effectively kills the engulfed microbes. It creates a microscopic chemical warfare zone right inside the cell.
That is absolutely astonishing. A highly effective strategy. What if MPO is deficient? Is that as bad as NADPH oxidase deficiency?
Fortunately, no. It's generally not as severe. While MPO adds a powerful weapon to the arsenal, MPO deficiencies are usually less clinically severe than CGD, NADPH oxidase deficiency.
Why is that?
Because even if MPO is missing, the hydrogen peroxide generated by NADPH oxidase alone is still pretty bactericidal. It shows the layers of defense. H2O2 can also be dealt with in other ways, partially reduced to the even more reactive hydroxyl radical, OHA, or fully reduced to harmless water by enzymes like catalase or glutathione peroxidase. So there are multiple pathways for handling these reactive species, both for killing microbes and for eventually neutralizing the weapons once the job is done.
Okay, let's switch gears again to another fascinating application of NADPH, nitric oxide NO synthesis. This molecule sounds like it wears many hats, involved in a whole array of biological systems.
It truly does. Its versatility is remarkable. Nitric oxide NO is a powerful free radical gas, and it's absolutely crucial to distinguish it from nitrous oxide N2O, which is laughing gas.
Right, different molecule.
Totally different. NO is incredibly transient and reactive. It has a very short half-life in tissues, maybe just 3 to 10 seconds.
That's short. Wow.
Yeah, because it rapidly reacts with oxygen and superoxide, getting converted into things like nitrates, nitrites, and peroxynitrite, another reactive species. This short lifespan is key to its role as a local signal. It acts quickly and nearby, then disappears.
And what are these diverse roles? Where does it operate?
Its functions are widespread. It's famously known as the endothelium-derived relaxing factor, EDRF. It causes vasodilation relaxation of blood vessels by acting on vascular smooth muscle, vital for blood pressure control and blood flow.
Okay, vasodilation. What else?
It also acts as a neurotransmitter in the brain and peripheral nerves. It inhibits platelet aggregation, helping prevent blood clots, and, as we hinted, it plays an essential role in macrophage function in the immune system, contributing to microbe killing. It's like a cellular conductor leading many different biological orchestras.
How is this remarkable, multi-talented molecule made? What's the enzyme, and where does NADPH fit in?
Nitric oxide is synthesized by a cytosolic enzyme called nitric oxide synthase, or NOS.
NOS. Okay.
The substrates for NOS are the amino acid arginine, molecular oxygen, O2, and, crucially, NADPH. NADPH provides the necessary electrons for the reaction. The enzyme then produces NO and another amino acid, citrulline.
Does NOS need anything else to work?
Yes, it's quite complex. It requires several important coenzymes, flavin mononucleotide, FMN, FAD, heme, and a molecule called tetrahydrobiopterin. It's a sophisticated machine for precise NO generation.
And I understand there are different types or isozymes of NOS, some always on, some inducible. How does that work?
Yes, that's key to its versatility. There are 3 distinct NOS isozymes, each from a different gene with different regulation and roles. We have 2 constitutive isozymes.
Constitutive meaning always present.
Exactly, synthesized at a pretty constant low rate. These are ELO, endothelial N and NO, neuronal NOS. Their activity is finely tuned by calcium levels. They are calcium calmodulin dependent. They produce low continuous levels of NO for ongoing physiological functions.
Like eNOS for blood vessels.
Right. eNOS in endothelial cells lining blood vessels mediates that continuous baseline vasodilation, keeping blood flowing smoothly. And NOS, mainly in neural tissue, acts as a neurotransmitter involved in things like learning and memory.
So those are the everyday low-level producers. What about the inducible type, iNOS? What makes it different? What's its role, especially in immunity?
The third is the inducible isozyme, iNOS. Unlike the others, iNOS is calcium independent, and it's normally expressed at very low levels. However, it can be massively upregulated, massively expressed in many cell types, including macrophages and neutrophils, as a powerful defense against pathogens.
What triggers that massive upregulation?
The key is that iNOS synthesis is significantly stimulated by pro-inflammatory cytokines like TNF-N and IFN-MO, and by bacterial components like lipopolysaccharide, LPS, signals that scream infection.
Right.
This stimulation leads to the production of huge sustained amounts of NO over hours or days. It's part of a robust, aggressive immune response, like switching from a gentle drip to a powerful fire hose of NO.
That brings us to how NO specifically causes vasodilation. How does that cascade work, from NO synthesis to relaxing blood vessels?
It's a really elegant pathway. Starts with ENO in the endothelial cells making NO. Because NO is a small gas, it rapidly diffuses across the cell membrane to the adjacent vascular smooth muscle cell.
Okay.
Once inside the smooth muscle cell, NO activates the cytosolic form of an enzyme called guanylyl-calase. This leads to the rapid formation of cyclic guanosine monophosphate, or CGMP.
CGMP, like CANMP, but with guanosine.
Exactly, a second messenger molecule.
So CGMP is the signal. How does that lead to relaxation?
The rise in CGMP then activates another enzyme, protein kinase G. This kinase phosphorylates calcium channels, which decreases calcium entry into the smooth muscle cell.
Less calcium.
Less calcium means less activation of myosin light chain kinase, an enzyme needed for muscle contraction. The end result is decreased smooth muscle contraction, which means relaxation, which is vasodilation.
Ah, okay. So NO, guanylyl cyclase, calcium kinase, protein kinase, G, less calcium relaxation.
You got it. And this is why vasodilator drugs like nitroglycerin work. They get metabolized to NO in the body, triggering this cascade, relaxing blood vessels, lowering blood pressure. NO is basically our body's own endogenous nitrovasodilator.
A natural blood vessel opener.
Exactly. And interestingly, under low oxygen conditions, hypoxia, nitrite, NO2, can also be reduced back to NO, which can bind to deoxyhemoglobin and get released, causing vasodilation specifically in oxygen-starved tissues. a clever way to direct blood flow.
That's remarkably smart. And how does NO contribute to macrophage killing of bacteria, linking back to immunity? You mentioned synergy.
Yes, it's a great example of immune synergy. Normally, iNOS activity is low in macrophages, but during infection, bacterial LPS and those inflammatory cytokines, IFN, TNFA, crank up iNOS production massively.
Okay.
Now, remember, the activated macrophage is also producing superoxide radicals, O2-EA, via NADPH oxidase during the respiratory burst.
Right.
These superoxide radicals combine with the large amounts of NO now being produced by iNOS. This forms reactive intermediates that rapidly decompose to produce the highly toxic hydroxyl radical.
Hydroxyl radical.
Even more potent.
Extremely potent and damaging. So it's a sophisticated tag team effort. NADPH oxidase makes superoxide, iNOS makes NO. They combine to make something even nastier to kill the pathogen. Beautiful interplay.
Are there any other key NO functions we should quickly mention?
Yeah, just briefly. As we touched on, NO is also a potent inhibitor of platelet adhesion and aggregation, working via that CGMP pathway again, crucial for preventing unwanted clots. And it functions as a neurotransmitter in both central and peripheral nervous systems. Truly versatile. We've covered a huge amount on the pathway and NADPH's roles. Now let's turn to what happens when the system breaks down, specifically with G6PD deficiency. What exactly is G6PD deficiency, and what are its main consequences?
Right. G6PD deficiency is a hereditary condition passed down genetically. It's primarily characterized by hemolytic anemia.
Hemolytic anemia and breakdown of red blood cells.
Exactly. And it's specifically caused by the inability of cells, especially red blood cells, to adequately detoxify oxidizing agents. It's a major weakness in the body's shield against oxidative stress.
How common is this? I know you said it's prevalent in some areas.
It's remarkably common globally. In fact, G6PD deficiency is the most common disease-producing enzyme abnormality in humans. It affects over 400 million people worldwide.
400 million? Wow.
Yeah, it's particularly prevalent in the Middle East, tropical Africa and Asia, and parts of the Mediterranean. It's an X-linked disorder.
Meaning on the X chromosome, so more common in males.
Exactly. More common and often more severe in males. And it's not just one single defect. It's really a family of deficiencies. There are numerous different mutations in the G6PD gene itself. Only some of these variations actually cause clinical symptoms.
What kind of symptoms might you see, especially early on?
A significant early sign can be neonatal jaundice, appearing maybe 1 to 4 days after birth. This jaundice, the yellowing, can be quite severe and is due to the rapid breakdown of red blood cells releasing bilirubin.
Okay.
Now, while severe forms can lead to issues like chronic hemolysis and maybe a slightly shortened lifespan, there's a fascinating evolutionary twist that helps explain why it's so common in those specific regions.
An evolutionary trade-off, like sickle cell trait.
Exactly like sickle cell trait. It's a classic example of natural selection. Despite the problems it can cause, G6PD deficiency provides significantly increased resistance to Plasmodium falciparum malaria.
Malaria resistant.
Yes, the most severe form of malaria. In areas where malaria has been historically endemic, individuals with G6PD deficiency were less likely to get severely ill or die from malaria.
So the gene, even with its downside, offered a survival advantage against a bigger killer.
Precisely. This selective pressure allowed the gene to persist to become common in those populations. It's a powerful example, just like sickle cell trait and also the thalassemias, which offer similar protection.
So G6PD deficiency affects all cells, but the worst problems are seen in red blood cells. Why are RBCs so uniquely vulnerable? What makes them the weak link?
This is a critical point, and it comes down to two unique things about red blood cells. First, RBCs rely exclusively on the pentose phosphate pathway as their only source of NADPH.
Their only source, no backups.
No backups. Unlike other tissues, say the liver, which might have alternative NADPH-producing enzymes like malic enzyme,
Right.
RBCs don't have those. If the PPP isn't working properly in an RBC, it's completely out of luck for NADPH. Its main antioxidant defense generator is offline.
Okay, that's huge vulnerability, number one. What's number two?
Number two is that mature red blood cells are basically just bags of hemoglobin. They have no nucleus, no ribosomes.
Stripped down for oxygen transport?
Exactly. This means they cannot synthesize new G6PD enzyme once they mature. So whatever deficient G6PD variant they start with, its activity will gradually decline as the RBC ages. They can't make more.
Ah, so older cells become progressively weaker.
Exactly. Older RBCs are particularly vulnerable to oxidative stress because their already limited enzyme supply has dwindled further. They have a fixed budget of enzyme that just runs down. with no way to replenish it.
That perfectly explains their unique susceptibility. Now let's trace the pathophysiology. How does this diminished G6PD and RBCs actually lead to hemolytic anemia? What's the chain reaction?
Okay, so diminished G6PD activity directly impairs the RBC's ability to make NADPH.
Right.
Less NADPH means the cell can't maintain the pool of reduced glutathione, GSH.
The antioxidant shield weakens.
Exactly. With less GSH, the cell can't effectively detoxify the constant barrage of free radicals and peroxides from metabolism or external triggers.
Yeah.
Also, GSH helps keep sulfhydryl groups on proteins, including hemoglobin, in their reduced state.
Okay.
When these sulfhydryl groups oxidize due to lack of GSH, the proteins, especially hemoglobin, denature and clump together into insoluble masses called Heinz bodies.
Heinz bodies, like little protein aggregates.
Yes, sticky clumps of damaged protein that attach to the inside of the RBC membrane.
And what happens to cells with Heinz bodies and damaged membranes?
The presence of Heinz bodies, plus further oxidation of membrane proteins, makes the red blood cells rigid. They lose their normal flexibility.
Can't squeeze through capillaries easily.
Exactly. These rigid, damaged cells are then recognized as abnormal by macrophages, particularly in the spleen and liver. These organs act like filters. They sense the inflexible, damaged cells and remove them from circulation prematurely.
And that premature removal is the hemolysis.
That premature destruction is the hemolytic anemia. The individual ends up with fewer, healthy, oxygen-carrying red blood cells.
It sounds like a tough situation for those cells. What typically triggers these hemolytic episodes? You said most people with the mutation are okay day to day.
That's right. Most are asymptomatic normally. Clinical problems, especially acute hemolytic anemia, are typically triggered by specific exposures that suddenly increase oxidative stress, overwhelming the cell's already weakened defenses.
What kind of triggers?
There are three main categories to know. First, oxidant drugs. The mnemonic AAA can be helpful for some common culprits. Certain antibiotics like sulfamethoxazole,
Okay.
certain amphomeriols like primaquine, importantly not chloroquine or quinine, and some antipyretics like acetanilide, but crucially not common ones like acetaminophen paracetamol.
So doctors really need to know about these specific drugs.
Absolutely vital. Prescribing one to an unknown G6PD deficient patient can cause a serious crisis.
Okay, what's the second trigger? You mentioned favism earlier.
Yes, favism. This is a specific, sometimes severe hemolytic reaction caused by eating fava beans, also called broad beans.
Just from eating beans?
Yes, they're common in Mediterranean diets where G6PD deficiency is also common. Not everyone with G6PD deficiency reacts to fava beans, but it's a strong link. Essentially, all patients who do experience favism have G6PD deficiency, often a specific variant called the Mediterranean variant. It's a direct gene-diet interaction.
Fascinating. And what's the most common trigger overall, something that affects almost everyone?
The most common precipitating factor for hemolysis in G6PD deficiency is, perhaps surprisingly, infection.
Infection. Why?
Because the normal inflammatory response to infection involves immune cells, like macrophages, generate a lot of free radicals, that respiratory burst we talked about to fight the pathogen. These free radicals, produced by the immune system itself, can diffuse into the already vulnerable red blood cells, causing massive oxidative damage and triggering a hemolytic episode. So even a common cold or a minor infection can become serious because the RBCs just can't handle that extra oxidative stress.
Let's talk about the molecular basis. You said it's a family of deficiencies. What causes this genetic diversity, and how does it relate to the different levels of severity?
Right. Almost all G6PD variants are caused by point mutations in the G6PD gene. Specifically, missense mutations, a single DNA-based change leading to a single amino acid change in the enzyme protein.
Just one amino acid difference?
Often, yes. And over 400 different G6PD variants have been found worldwide. This huge genetic diversity directly explains the wide spectrum of biochemical and clinical outcomes we see. These single amino acid changes can impair the enzyme in different ways, maybe decrease its catalytic activity, making it slower, or reduce its stability, making it fall apart faster, or alter how well it binds to NADPH or glucose 6-phosphate. Mutations near where the enzyme subunits join together can particularly affect stability since G6PD works as a dimer or tetramer.
So the severity depends directly on how much functional enzyme is left in the red blood cells. Can you give examples of the different classes?
It's exactly. Clinical severity correlates directly with residual enzyme activity. They're often classified. For example, class 3 variants, like the common G6PDA, are considered moderate.
Moderate deficiency.
Right. The enzyme might be unstable, but work okay kinetically when it's there. So activity is higher in young red blood cells, reticulocytes. Older RBCs lose activity faster and are the ones mainly destroyed during a hemolytic episode. But because new cells are constantly made with some activity, these episodes are often self-limiting. The body can recover.
Okay. And the more severe variants?
Then you have class 2 variants, like G6PD Mediterranean, which cause a more severe deficiency. Enzyme activity is significantly lower overall, and class I variants are the rarest and most severe.
Class 1.
These lead to chronic non-spherocytic hemolytic anemia, meaning red cells break down constantly, even without any specific oxidative trigger. It's a persistent, lifelong anemia.
That's a huge difference based on potentially just one amino acid change. Has anyone ever been found with zero G6PD activity?
That's a really telling point from the source material. Large deletions or frame shift mutations that would completely abolish G6PD activity are basically never found in living humans.
Meaning?
It strongly suggests that a complete lack of this enzyme is likely lethal. You simply cannot survive without any G6PD activity. It underscores its absolute, non-negotiable essentiality for human life.
So, bringing this all together for you, our listener, what does this all mean? We've journeyed deep into the pentose phosphate pathway and this remarkable molecule, NADPH. We've peeled back the layers to see how this pathway, without using any ATP directly, is utterly indispensable. It builds the fabric of our genetic material, synthesizes crucial hormones and lipids, and acts as the body's frontline defense against cellular damage and microbial invaders.
That really covers a lot of ground.
And we also saw how a defect in just one key enzyme, G6PD, can ripple through all these vital systems, leading to a common and complex clinical condition that actually teaches us a huge amount about human health, evolution, and adaptation.
It's truly a testament to the incredible complexity, but also the elegance of our internal biochemistry. These aren't just abstract reactions, they're profoundly interconnected processes. Every molecule, every enzyme playing a critical, often hidden role in keeping us healthy, keeping us alive.
Yeah.
From fighting oxidative stress to powering immunity and building the molecules of life, NADPH and the PPP are working silently, constantly behind the scenes.
So maybe consider this, how the body's intricate balance, which seems so stable, can hinge on the precise function of just one single enzyme, like G6PD, and how even tiny changes, like 1-point mutation, can dramatically shift that balance.
It's quite profound when you think about it that way.
This deep dive into the pentose phosphate pathway reveals not just, biochemical elegance, but a really deep story of adaptation, of vulnerability, and this constant unseen dance between creation and protection happening inside us all the time. It makes you wonder, what other hidden pathways might be shaping our health and disease in ways we're only just starting to appreciate, pathways just waiting for their own deep dive?
That's a great question to ponder.
We really hope this conversation has brought you a step closer to mastering these fundamental processes, unlocking that deeper understanding of the amazing biological systems that make us who we are.